Tendons connect our bones to our muscles. This occurs gradually. When the functionally graded tendon-to-bone transitional region is damaged or severed, medical attention is necessary. Doctors and clinicians using current surgical and medical treatments have inefficiencies in repairing the tendon-to-bone interface once severed. Bone tissue thrives under compression, while tendon tissue develops under tension. For this reason, there is a material mismatch that occurs at the interface between tendon and bone. Clinicians have noted that approximately 30 to 90% of tendon-to-bone insertion site repairs fail. Engineers and scientists have postulated that this is the result of the material mismatch between tendon and bone and strive to fix this problem. Engineers have been working on creating synthetic scaffolds to assist clinicians in repair. One way to fix the broken tendon is to use a fabric scaffold, much like the fabric in your clothes, to replace the tendon. However, these scaffolds often fail due to fiber-on-fiber or fiber-on-bone wearing. Engineers at the University of Memphis, affiliated with the Orthopedic Research Society, working in the field of regenerative medicine, are therefore searching for ways to incorporate cells and cellular materials to improve the long-term viability of synthetic grafts using our novel bioreactor system. First, we add cells to the scaffolds. Bone cells, called osteoblasts, are seated onto the right-hand portion of the scaffold. This will be the hard, bony portion of the scaffold. Fibroblast cells that secrete elastic matrix serve as the tendon-like portion of the scaffold and are seated onto the left portion of the scaffold. The bioreactor chamber isolates the osteoblast side from the fibroblast side using a clamp. Because the two scaffold portions are isolated, we have been able to exercise the fibroblasts inhabiting the left side of the scaffold via stretching, while allowing the osteoblastic side to remain unstretched. A biological addition to a synthetic scaffold for enthesis or tendon-to-bone repair could serve to decrease wear trends, increase strength, and gradually transition from tendon to bone. The bioreactor mimics the kinds of motion that the tendon sees in the body and makes it behave more like a tendon tissue. We found that fibroblasts grow best under repetitive stretching and with constant medium perfusion. In fact, we found that exercising the fibroblasts resulted in 2 times the matrix production as compared to fibroblasts that were not exercised. When we exercise fibroblasts that are growing in the same chamber with osteoblasts, the osteoblasts also deposited more matrix components. This project uses engineers to create a bioreactor, scientists to evaluate cellular responses of bone and tendon, and clinicians to make the improved treatment a reality.